Hi, and welcome to Psyched YouTube channel. My name is Michael, and I'm here with my friend Mile. How you doing, man?
Hi there, Michael. I'm good. And today we're not alone because today we are with Dr. Ali Salehinejad, which is a colleague and friend of mine. And we recently published an awesome study, and that's the one where we're going to talk about today. So, Ali, how are you doing?
Hi, Miles and Michael. Thanks for having me. I'm good, and I'm happy that I'm here in this video.
So, what are we going to talk about? What study did you do?
I think the topic of the paper was it reads cognitive functions and underlying parameters of human brain physiology are associated with chronotype. But in brief and summary, what we wanted to study was to see what is the relationship between chronotype and human cognitive functions and some underlying parameters of human brain physiology that support those cognitive functions. So this is what we studied. And by chronotype, I mean the circadian preference. Chronotype is an individual difference in human beings. So it means that how much you are a morning person or evening person or neutral. Because we have two major type of chronotype, which is kind of morning person or early bird, or evening person or late O. And the important thing is that this chronotype has a normal distribution in the society, so which means that a lot of us are affected. I'm personally an evening type, which means in the morning I'm not very alert, but in the evening I can concentrate better. So we just wanted to see how this affects human brain and cognitive functions.
Right. Yeah, I do remember that you are more of an evening type, because when you started this study, that's the time when I was also at your institute. By the way, you work at the IFADO in Dortmund in Germany. I forgot to mention that. But when I was there, you started your study, and I remember that you said that you had to be there early in the morning, and you really didn't like that. Did that change at all? Are you now more of a morning person, or?
Chronotype, of course, is affected by age. So the older you get, the more morning type you will be. I mean, at least, it means this is what the data shows. And for me, in the beginning, it was very hard because I had to start experiment around 7:30 in the morning, prepare everything, which was very early for me. And also one time in the evening. In the beginning, it was very difficult, but I think in the middle of experiment, I got used to it because I just regulated my sleep time. So this is something that you can force yourself to change your sleep habit, so you can get up early in the morning. But still in the morning, I was not alert, you know what I mean? So I just needed coffee for sure.
Well, that's true for a lot of academics. So, but it's already good to, like, this is the first go home message. Whatever we're going to talk about today, there are ways that you can deal with that, right? So it's not like that if you're an evening type, then you're screwed for the rest of your life, but you can actually do something about it. All right. So I guess what we want to do today is basically discuss the paper that you wrote, because I think there are some really interesting things in there that are interesting for every one of us. But there will be also some more difficult things, which where we can explain how the neuroscience behind something like this works. So I think what I will do in this video is that I will just show the pictures on screen, or not the pictures, the figures on screen, and then by that we can basically go through them. So why not just start with the first figure, which is figure 1. And figure 1 is kind of like an overview of the entire study that you've done, right?
Yes. Can I just add a bit explanation before we go to figures?
Yeah, sure.
Chronotype and circadian preference is a topic which is mostly studied by people who are expert in sleep and circadian rhythms. I'm a cognitive neuroscientist, and what was important for us was to see what happened to human brain and cognition under chronotype effect. So this was what motivated us to study. Of course, chronotype and circadian preference has a lot of effect on the body physiology, metabolism, cell metabolism, and different things. But my point was that we look at this variable from cognitive standpoint to see how human cognition and those parameters that are important for our everyday cognitive function are affected. And now I think we can go to the figure 1. So I explain about what we did.
Awesome. Yeah. So if we take a look at figure 1, then what the first thing that comes that we see is that there is a distinction in 3 kind of different parts of the experiment, right? Cortical excitability, neuroplasticity, and brain and cognition. And that's already what you said, right? There are effects on the physiology of the brain, so basically how active the brain is, but also on the behavior and cognition, so how well you function.
Yes, exactly. The idea was to see if how pervasive is the effect of chronotype, because if you can see a pattern from the basic physiological parameters then to behavior and cognitive functions, then I mean like a hierarchy, then if you see an effect from basic physiology to behavior and cognition, then you can claim that there is something there. So yeah, we started from monitoring parameters of human brain physiology, that these parameters are important for everyday function like brain excitability and also neuroplasticity. These are some physiological phenomena that happen in the brain that support our cognitive function. And besides these parameters, we also wanted to see what happened at behavioral level. But when somebody learns a task, when somebody is asked to memorize something in the morning or in the evening, and yeah, so it was 3 major parts. This experiment has 3 major measures: brain excitability, neuroplasticity, and cognitive function. But cognitive function has also 2 subdivisions: so behavioral performance and electrophysiological correlates of the brain.
Right. And we will, of course, talk about all of those.
Yeah.
So basically, just to give the basic structure, you had 32 participants, right?
Yes.
And half of them were identified more as a morning type, and the other half was more an evening type, right?
Evening type, right.
And then each participant was tested in the morning and in the evening. So basically you had a morning type that would be tested in his preferred morning or her preferred morning, and then in the not so preferred evening, whereas an evening person was also tested in the morning and the evening.
Exactly. So each participant attended 10 sessions.
Mm-hmm.
5 sessions were in the morning at 8:30. The beginning of the experiment was at 8:30, and 5 other sessions was at 7 p.m. So this was fixed for all of the participants, for all 32 participants. So this means that every participant did the same task or was measured with the same physiological measurement in the morning and in the evening. So those who were morning types were measured in the morning, which we call circadian preferred time for them, and those who were evening were measured in the evening, which is the circadian preferred time for them as evening time. But they were also measured in the circadian non-preferred time, which is morning for evening types and evening for morning types.
Yeah, and just to emphasize, 10 sessions. That's a lot.
That's a lot, and there was one week interval between each session at least. So this, I think, the data collection took at least 1.5 years.
Yeah, 1.5 years, and basically you have 320 sessions. That's a crazy amount. So
you just skip those test sessions and those sessions that are canceled. So yeah.
Of course.
Yeah, I mean, yeah.
Yeah, that's what a lot of people don't realize is that, when you're testing with real life participants, then there's a lot going wrong with planning and stuff like that. Yeah. So if you have to do 320 sessions, you probably have to plan 500. So, but okay, let's, so basically let's get into it. Let's start with the cortical excitability. And, I think we can, I mean, if it's okay with you, jump then to figure 2. But before we jump to figure 2, I think there is some explanation necessary, because what you did to look at cortical excitability, and cortical excitability is basically, if you would translate it into very easy words, is how active is the brain. You did do this by using transcranial magnetic stimulation, or TMS.
Yes.
And TMS is basically a magnet that induces an action potential in an area that you stimulate. And you stimulated the motor cortex, so you try to induce an action potential in the motor cortex, right?
Right. So just if I want to also put cortical excitability from the way we looked at it, what you say was completely right, but it's also, we can also very, very easily say the cortical excitability is how the brain processes an input from the environment. And this is always the result of two basic processes, like in cortical inhibition and cortical facilitation. So there should be always a balance between. Yeah, this is what we measured, as you said, with TMS. So in TMS, there are also, with TMS, you can use different protocols. And by different protocols, I mean that you can change if you apply one magnetic pulse or two magnetic pulse, or how long is the interval between these two magnetic poles. I don't want to go into details, but with these TMS protocols, you can measure different parameters of cortical excitability, which I said one of them is cortical inhibition, the other one is cortical facilitation, another one is corticospinal excitability or corticospinal facilitation. So we used four different protocols that are commonly used in TMS studies. And the idea was to monitor cortical excitability when people are at the circadian preferred time and when they are at the circadian non-preferred time. And what we found, this figure 2 shows that we have 4 subfigures, A, B, C, and D, and all of these figures shows that when people were at the preferred time, for example, morning type in the morning and evening type in the evening, and they showed higher cortical facilitation and lower cortical inhibition. So this means, from physiological standpoint, this means that their brain were more, I would say, ready for processing or more equipped or prepared for process inputs from the environment. So this is how we can translate it very easily.
Right. Yeah, and that's really cool, because basically that is kind of what you would expect if somebody would describe him or herself as a morning or evening type. Then kind of what they describe is, I don't feel like, if you're an evening type and you're in the morning, then you're just a little bit sluggish and you don't feel ready yet. And it's kind of you see that in the physiology.
Yes, for example, one of the things that people who are evening type is, okay, in the morning I cannot learn. For example, if I have to go to mathematics class at 8 at 8 a.m., I cannot learn. So learning is something that we might know that it happened. In brain, what happens when we learn something, it's always about these physiological parameters. So if we can learn something, it means that our brain excitability is ready enough to process an input, then we can learn and learn that item. So these parameters are the underpinning of the learning and other cognitive process.
Right. So yeah, maybe let's go a little bit more into detail in a few of those subfigures. So you already said that basically with TMS you can measure facilitation or excitation and inhibition, and basically how you can think about the brain is that the brain is a network that has neurons that are more excitatory and more inhibitory, right? And excitatory means just they are activating the muscle, whereas an inhibitory signal is more something that suppresses the muscle, that refrains you from being active, basically. Right. So, and when we go to figure 2A, for example, this is when you give when you look at the response to a single TMS pulse, right? So you basically have this TMS machine on somebody's head and you give one pulse and then you see how somebody's muscle reacts. Right. And that typically tells you a little bit more about this balance between excitation and inhibition. But yeah, we typically say the higher the response, the more excitable the brain is, right?
Right. So in this, in subfigure A, it is called IO curve protocol. So in this protocol, we apply TMS at 4 different intensities, and the higher the intensity, we expect the higher the motor evoked potential, because we measure motor evoked potential. I hope these terms are not very, very specialized, but you can always restate.
Right. Yeah. So a motor evoked potential is just a response of the muscle to a TMS pulse.
Yes, and so when we apply TMS on the brain of people, for example, on morning types, in the morning, their muscle activity was significantly larger in the morning than in the evening. And the reverse pattern was true for evening type, which means that when we apply this protocol on the head of evening types, the muscle activity was significantly larger in the evening, which means their cortical spinal excitability was high, because this subfigure specifically is sensitive for cortical spinal excitability and is related to a neurotransmitter system which is related to excitability, which we call glutamate. And in higher intensity, the glutamates neurotransmitters are involved.
Right. So what you're basically testing is how easy a signal from the motor cortex can travel to a muscle. You're basically doing this by applying TMS. And what you see is that in the morning, that is easier for people who are morning type, and in the evening, that is easier for people who are an evening type.
And you were able to do that by applying a magnetic pulse to the motor cortex of the brain, I presume?
Yes.
Right. We apply magnetic pulse on the motor cortex, left motor cortex, and then measure muscle activity of the right hand.
So just by applying this magnetic pulse, you saw that there was a motor response, saw twitching of hand, I guess, or something, finger.
Yes, in this muscle, which we call ADM muscle, in different labs they use different muscle, but in our lab in IFADA we use this muscle. The thing is that just imagine that I applied this TMS on your head one time in the morning and one time in the evening. Then I compared what are the size of your muscle activity in the morning and in the evening. Then if you're a morning type, your muscle activity is expected to be larger in the morning. Of course, you could be outlier and doesn't show this pattern, but this was what we found at group level. And
that shows them that among morning people, their brain was more excitable in the morning, and evening person the opposite.
I could also say their brain was more prepared to be excited to process information, because but the way you also put it is right. Yes, their brain was more excited in the morning.
Right. So that's really cool. And then basically what you're doing in figures B, C, and D, we don't have to go into detail in those, but you're basically kind of looking separately to the excitation part, the facilitation part, and separately to the inhibition part, right? And then basically you see that facilitation is stronger when it fits their chronotype, and inhibition is weaker when it fits their chronotype, right?
Yes, because figure A was just excitability. I just like to briefly talk about figure B, which includes for both cortical inhibition and facilitation. Again, in this protocol, we can measure cortical facilitation and inhibition. So for the facilitation, we found the same pattern of figure A. So when people were at their preferred time, their cortical facilitation was significantly higher. Like morning types showed higher cortical cortical facilitation in the morning, and evening types showed higher cortical facilitation in the evening. But about the cortical inhibition, they showed significantly less cortical inhibition in their preferred time. So it means that when you are at your preferred time, your brain is less inhibited. Your cortical inhibition was less. This is what we found.
Right. So when you think, of course, what you said in the beginning about this balance between excitation and inhibition, you could basically say this is biased towards being excited, being active, being ready to do something, right?
Yes, exactly. Right. Because later I show you in the behavioral results that, for example, motor learning, that when you learn a pattern of a sequence learning, so when this happens, that there is a balance between excitability and inhibition, and usually when there is less inhibition, less cortical inhibition, this learning happens based on some data. So I think this physiological pattern show us that we can expect that the behavioral performance would be also different in the morning and in the evening.
Right, that's really cool. So you already mentioned motor learning. We will come back to that later on. Yeah. When we talk about learning, we talk about we typically talk about, when we talk about brain function, about neuroplasticity, right? So when you learn something, there's neuroplasticity going on. There's wiring between neurons that becomes stronger. Now that is another thing you looked at, and that's basically, if we go to the next figure, if you're ready for that, yeah, then that's kind of what you looked at with TDCS, transcranial direct current stimulation. And that's kind of, so basically coming back to this, the previous figure is basically just the excitability of the brain in that moment. But then in figure B, you looked at whether this effect is true over a longer amount of time, right? So whether this effect is, whether there's a plastic change that remains for a while.
Yes, exactly. You know, in the first figure, we found that the brain is more excited or more excitable. Then in the second figure, what we wanted to do, we said, okay, what if we now induce plasticity in this state of the brain? Because brain is now more excited. So what happens if we induce plasticity, neuroplasticity in the brain? So we expect that, okay, if the brain is more excitable, we expect that we see a larger plasticity induction. So again, as you put it, neuroplasticity is another physiological mechanisms underlying learning, especially learning and memory. So which means that if you can have a larger plasticity response in the brain, your learning or your cognitive functions is expected to be also enhanced. So in this figure, in the second figure, we applied two brain stimulation, non-invasive brain stimulation protocol, which we with TDCS, transcranial direct current stimulation. So one of these stimulation induces we call LTP-like plasticity. I hope it's not very, very specialized term. And the other one induces LTP-like plasticity. So basically, it means that it can enlarge the muscle response that we already talked about. With the LTP-like plasticity, we can enlarge the motor response, the muscle response, and with the LTD plasticity, we can reduce the muscle response. But what is important, again, here, there is a balance between LTP and LTD in order to learning happen. So we applied TDCS again in all of the participants in the morning and in the evening. So each participant received 6 sessions of TDCS, anodal and cathodal and sham TDCS. Sham is the control condition in the morning, and the same protocols in the evening. And basically, what we found, our expectation was true. So we found that when you are at your circadian preferred time, your brain can induce larger plastic response, which means morning types had larger plasticity induction in the morning, and evening type had larger plasticity induction in the evening. So this is basically the summary of the second figure.
Right. Yeah, that was quite a lot, and it's really cool. So just coming back to the LTP and LTD thing, so that stands for long-term potentiation and long-term depression. And kind of those terms already say it, right? There's a potentiation, so something is made stronger, whereas a long-term depression is something that is made weaker, right? And you use TDCS in this case, and you apply TDCS over the motor cortex. And I think what is important to mention is that TDCS is a technique that is around for, 20 years? So there's a lot of research behind the idea that if you use the so-called anodal TDCS, that basically increases the responses of the muscle with this LTP-like mechanisms. Whereas if you apply cathodal TDCS, that is more likely to decrease the muscle response, right?
Yes, it's somehow related. I mean, close to those facilitation and inhibition that we talked in the first figure. Mm-hmm. Yes, TDCS. With TDCS, we apply a very weak direct electrical current to the head, and then we can either measure a motor response or also cognitive and behavioral performance. Mostly TDCS is used applied over the head to for measuring also cognitive functions, because you can apply and then see how people, for example, do a working memory test.
Right.
Yeah, this is what we did. This was the most time-consuming part of the experiment, because every participant had to come 6 times to the lab, and you know, it's sometimes also ironic that this amount of work is just shown in one figure.
Right.
It's almost the summary of, I would say, 8 months or something, even more. But the good thing is that we found a converging finding. We found that, okay, when the brain is more excitable at your circadian preferred time, then you can also induce larger change at your circadian preferred. This figure has some clinical and also public implications, because when we can induce larger changes at some specific time of day, so it means that in some patients who has a plasticity deficit, patient with stroke, then it has some implication. But also it means that you can learn materials better in the morning if you are morning time.
Right. So just to even simplify it a little bit more. So why were there 6 sessions? Because you tested each person in the morning and in the evening, and of course you had the morning and the evening type, and you did apply the anodal TDCS, which should increase the activity of the motor cortex, the cathodal TDCS, which should decrease the activity of the motor cortex, but also you did a sham paradigm. So that's basically like a placebo control, right? So in science, we always strive to have a placebo control, and that's what you did as well. So I just found it important to mention that, because, you know, the placebo control is really important.
Yes, otherwise you cannot say this effect is really because of placebo or not.
Right. And just to summarize it, a morning person shows bigger responses after this excitatory protocol of anodal TDCS compared to when you would have that same participant tested in the evening, whereas an evening person shows a bigger facilitation after anodal TDCS in the evening if you would compare it to the morning. And then for cathodal TDCS, it's the opposite.
Yes, right. I like to give an example, which probably is easier. Just imagine when you go to gym, you do some practice to strengthen your muscle. Just imagine if you do this practice in the morning, then this has a larger effect on your muscle size compared to the evening. So basically, if we translate to the brain, this graph shows that yes, your brain is more ready to be changed in the morning if you are morning type, and is more ready to be changed in the evening if you are evening type. Yeah.
Right. Yeah, and that's really cool. So that and that's of course what you said. That's not only clinically important, but you basically to everybody in everyday life that's important. So that's really cool. So in this case, in this paradigm, you basically induce neuroplasticity by using TDCS. But of course, we mentioned it before, neuroplasticity, we can induce ourselves as well by learning, right?
Yes, right.
So, and that's what neatly fits to basically the second, the third part of your experiment, where you also looked at motor learning, right?
Yes. So far, our measure was limited to physiological measures, which are based on measuring excitability of motor cortex. But behavioral paradigm is a very well established paradigm in psychology and also in science. Okay, if these are the effects, our question was that what happened at the behavioral level? So if this is the pattern that you see at brain level, then you expect to see changes at behavioral performance when you compare performance in the morning and evening. So motor learning is a behavioral task that is that measures implicit motor learning. It's important to mention that when you learn, in this task, participants should press some buttons, based on a pattern that could be random or non-random, and basically this task measures motor learning. But what is important to mention here, when learning happened in this task, some MRI studies and MRS studies shows that what happened in the brain is very similar to when you apply TDCS. So in the previous figure, we showed that when you apply TDCS, there is different results in the morning and evening. So we asked participants to do this task one time again in the morning, exactly at the same time that we applied TDCS, 8:30 in the morning, and also one time in the evening. And then we compared the behavioral performance. So with figure 3, with figure A and B, which is for early chronotypes and late chronotypes, this figure just shows that when you are morning type, you learn this sequence significantly better in the morning, and when you are evening type, you learn this sequence significantly in the evening. And this was associated with the electrophysiological correlates of learning, which we measured with EEG. I can go into details, but I think maybe it's not needed.
Yeah, so let's just quickly summarize it. So the task that people had to do is kind of they saw a sequence, and they basically with their fingers had to follow that sequence, right? So it's kind of like learning a new piano piece, and you have to follow the right orders to get the right tones in the right order, right?
Right. You should respond to the sequence that you see as fast as possible, but also correct. Right,
and that's easier for a morning person in the morning, and it's easier for an evening person in the evening. Basically, that's what you're showing in this figure, right?
Yeah, this curve, the steeper the curve, which means that the higher the learning. So this means that the morning people could learn this sequence implicitly, significantly in the morning, and evening types could learn this sequence significantly better in the evening.
Right, and then, I mean, for people who are interested, who are looking very specific at these figures, you see then in BL6, which is block 6, that there is this basically it goes back all the way top. That's one block where you show them a random sequence again, right?
Yes. So the previous block, except block 1, they have a sequence. I mean, there is a pattern, but it's implicit. You cannot guess. But in the block 6, suddenly the sequence is random. So the difference in the reaction time between block 5, which is the previous block, and block 6 is indicative of motor learning. And the larger this difference is, the better is the learning. So this is what we found.
Yeah, and that's really cool. And that's just basically how this task is typically done in the literature. That's why you see this response. And also after this block 6, you see that people return more easily back to their good performance. So you could basically say that block 6 is kind of like a distraction, and people.
So I didn't want to go into detail, but yes, the difference between block 5 and 6 is called learning acquisition, and the difference between block 6 and the next block is learning retention, which means again the learning pattern was reserved, was kept when again the sequence was the same.
Right. Yeah, and then you measured the brain physiology at the same time. So basically, how you did this, people had a EEG cap on. So EEG basically measures your what we colloquially would call brain waves, and you're basically looking at the brain waves at the point that participants doing this task, right?
Right. With EEG, you can measure these brain waves, and when participants were responding, we could also measure what happened in the brain, and which we called event-related potential. I think this is your expertise. You can explain it much better. Yeah. So these event-related potential have different components, and one of the components is P300, and which reflects that when your brain is processing the stimuli. So we found again that when people were learning, when morning types were in the morning, this component was also larger, which was associated and correlated with the behavioral performance. So this basically confirms our behavioral result.
Right. Yeah, it's interesting that you say that this is my expertise, because I do recognize those figures as.
Yeah, I think these figures were designed, of course, by you, but your PhD your PhD project was full with ERP measures, as far
Right.
as I know.
Yeah. So I think just to, without going into too much detail, so the P300 that just means positive peak after 300 milliseconds. And this is basically what you can see in the gray area. And just, yeah, EEG scientists are really weird because they always flip the, they flip the y-axis. So the positive peak is going down and the negative peak is going up. So in this case, this thing that goes down is the positive peak 300, P300. And you can see, if you just ignore the dotted lines, if you just look at the full lines, you can see that in the figure C, the red line is smaller than the blue line, and in the other figure, it's the opposite way. Again, confirming this idea that it depends on whether you're a morning type or an evening type, how your brain basically reacts to this task.
Right.
All right.
So I mean, so the way I interpret it is you've gathered evidence now from several different sources, right? You have behavioral data, you have electrophysiological data, you have from neuroplasticity, and they all indicate that this distinction between morning people and evening people is not something that we make up. There's actually actual data that supports this to be true. The learning is easier in your preferred time of the day.
Right. I think this is a very important point that you mentioned, Michael, because in most studies we usually have one or two measure, and always the reviewers say, okay, it's better to have a different measure so you can confirm your results. But when we see a pattern at different level, from basic physiology, cortical excitability, neuroplasticity, and then behavioral performance, then you can say, okay, there is something there. Still, in science, you can never say it's always 100%, but at least we can say that we found a converging pattern at different level, and then it's more reliable compared to when you have only one single piece of data.
Right.
Yeah, and I think that's really important that you mentioned it, because most studies would just take one figure or basically one part of the experiment and do that and then publish that, whereas you did basically everything, and we're not even done yet. And that's really cool, and it's so nice that it's converging evidence. And more importantly, you show this within the same group of participants, right? So you basically get those participants 10 times in, and you do all these different measures in the same participants, which is way better. You can, of course, always compare a single person to himself or herself better than you can compare it to another person. Basically meaning that your statistical precision is also a lot better.
Yes, thank you for restating it much easier.
Yeah. So and of course, what I didn't mention, and I should have mentioned this in the introduction, but of course, this is published in Nature Communications, which is part of Nature, which is one of the two biggest scientific journals there is for our field. So, there's a reason why it's in there, right?
Yes, when I think one of the reasons is that when you have a lot of data, then and you want to merge and publish put together, then you have a higher chance to publish in higher journals, better journals. Right.
So what we looked at is how excitable the brain is, how easy it is for the brain to learn, and we looked at this by looking at neuroplasticity and by motor learning. But of course, this is all related to movement, right? You also looked at more cognition, so how more thinking power, right? So memory, attention, this kind of stuff you look as well at.
Yes, and the title of this paper starts with cognitive functions. So cognitive functions and underlying brain physiology are associated with chronotype. So basically, the last piece of measures that we had included higher order cognitive functions that we need for not only everyday functioning, but also for these are also the functions that are mostly impaired in some neuropsychiatric disorders. So working memory and selective attention and sustained attention. So the idea was that, okay, if we see that the brain physiology and also motor learning are different at different time of the day for different chronotypes. So what about the higher order cognitive functions? Of course, as you said, these tasks are not related to motor cortex, but human motor cortex has usually been a model for studying human cognition, because in the motor cortex you can have more visible and objective responses. So in this figure we ask participants to do 3 cognitive tasks: NBAC task, which is a working memory task; the stroke task, which is a classic task and measure selective attention, also interference, but mostly known for selective attention; and another task which is called AXCPT, which measures sustained attention. So again, all of the participants perform these tasks in the morning and also in the evening, one time in the morning and one time in the evening, while their EEG was recorded, because we also wanted to see the electrophysiological correlates. And in this figure has 9 subcomponents. The first 3 A, B, C show that the performance in working memory was significantly better. Again, when you are at your preferred time, morning types had better working memory in the morning, and evening types had better morning working memory in the evening. And this was confirmed with their EEG data. So they had a larger P300. And figure D, E, E, F, the next 3 subcomponents are about selective attention. It showed the same pattern. The selective attention was significantly better in the morning for morning type and evening for the evening type. And the last task also showed the same pattern. It was about sustained attention. I can go into details if needed, but I think the pattern, is clear.
Right. And so basically what you're showing in this part, and, I kind of feel bad for just talking about it so shortly, because this is, as you say, it has 9 subfigures. You did 3 tasks, no participants. This is a lot of data, but exactly. It can be summarized quite quickly by saying even your cognitive performance is also relates to your chronotype. So if you're a morning person, you're cognitively better, meaning you have better attention, you can attend to things better, and your working memory is better. Working memory, by the way, is just a form of short-term memory where you also manipulate the things you keep in mind. So, for example, if you have to sum the numbers 3, 4, and 5, then you have to remember the numbers 3, 4, and 5, which is your short-term memory, but you also have to sum them up. So you have to do something with those numbers. That's an example of a mathematical task, but it's also involved working memory. So,
working memory was, sorry for interruption, but working memory is quite important because it is considered, as you know, is a very fundamental cognitive function, because it underlies a lot of cognitive function, and also some people believe that it's correlated with also intelligence, right? Because it shows how long you can keep items in your memory, and in order to have better working performance, in order to have better performance in the school, so you definitely need a good working memory.
Right. Yeah, definitely. So and that also shows that intelligence, of course, working memory, there are a lot of studies that relate to intelligence, that that isn't a fixed thing. It really depends on external factors, right? Yes, if you, it's also related to attention. Of course, these measures will also be related to each other, right? If you have more attention, you will also be better able to perform a working memory task.
Exactly.
Yeah. And then, quickly summarizing, as you said, then in figures, the figures on the right, you can see that is basically again confirmed by the electrophysiological measures with bigger responses where you would expect them. So, it's kind of like, if you confirm your hypothesis, it's always like, we did find what we wanted to find. So, and then there's nothing to too much discuss anymore, right? So,
Yes, as I said, the important thing was that we found these pieces of evidence at different levels. So
Right.
this makes the results, I think, at least reliable at this stage. Of course, we need more investigation. But what I wanted to emphasize one more time, in the beginning of our talk, we talked about cortical excitability, which I said I would define as the cortical response to processing an input. So
Right.
these tasks exactly shows our behavioral counterparts of cortical excitability, because you see a stimuli on the screen, then you have to respond to it. And that's why, I also did some correlations between the cortical excitability and this cognitive task performance. There were some significant correlations too, which are important. And my point was that these tasks are the behavioral counterpart of those physiological parameters that we measure. Because at the end, we want to see what happened to these cognitive functions. We cognitive neuroscientists, we want to see what happened to these cognitive functions in order to enhance them, or if you have some clinical preference, in order to improve them in some patients who lack these cognitive functions.
Right. So you mentioned clinical work a lot, and I know you are involved with that as well. Can you give some examples of clinical populations for which this research is really important?
Yes, I mean, in the discussion, I thought I speak about some clinical conditions, but I think one quite relevant condition is those patients who have plasticity deficits, and the most common case I would say is stroke patients. Mm-hmm. So when you have a stroke, then you have some movement deficits. You cannot move your hand or your body good enough because of a stroke. This means that there is a plasticity problem. So people in clinical neuroscience and also clinical psychology try to use this non-invasive brain stimulation, or even some other methods like deep brain stimulation, which is another but invasive way of brain stimulation, to restore this plasticity. Because if you can restore this plasticity, then you can enable those people to walk again like before, or improve their movement. So those who have plasticity problems and movement problems are one of those groups that can benefit from this result. But we have also some neuropsychiatric conditions that these patients have. Their problems is also related to chronotype, like attention deficit hyperactivity disorder. These patients are usually evening type. I mean, they're usually
okay.
You cannot claim, but the rate of evening type is high. Or in bipolar disorder, there is always circadian problems. And this data also shows that if we can regulate their circadian rhythms and or apply interventions, those interventions that are based on brain plasticity at their circadian preferred time, then you can expect a higher or more effective therapeutic response.
Right. That's cool. So, of course, what we talked about now is that this is really important for neuropsychiatric or neurological disease. But I guess this is also important just like for everyday people, right? I mean, what does this say about the general 9 to 5 work days? I mean, if we follow this data, then more flexible work hours would be better, right? Where people can, if you're a morning type, start earlier in the morning, whereas if you are an evening type, it would be more efficient to work later on the day, right?
Exactly. In 2020, last year, there was a paper published in Nature Human Behavior. So they measured the chronotype in school-age children, and then they correlate the chronotype with the performance. And the results shows that, I also cited this paper in my paper. The results show that those students who are morning type, who are evening type, they have worse performance or weaker performance in the morning. So at least at the school level, this is shown by some correlational data. But about the thing that you said, yes, I think this means that we can have flexible working time. And this was also a hot topic for some time, I think, in Europe, because, you know, in each year you have to change the time zone in 2 different times. And this, for some people, it's really very, very demanding if you change your working time. Well, out of sudden, there is 1 hour change. But basically, this data suggests that people should be able to choose to work when they have, when at least when their brain is more prepared. And I think this pandemic was a good experience, at least that showed that when people work from home officing, and maybe when they can work at their preferred time, maybe they could be as productive as when they have to come in the morning. So yeah, it has broad implications for public health and society, which I discussed in the discussion.
Right. So a very important paper. Yeah, I mean, all my compliments to it. Michael, do you have anything else you want to ask or say?
Yeah, I mean, so I've been relatively quiet in this interview. I must have been taken back to listen to you guys talk because, you know, you guys are scientists. I'm not really quite there yet, but I'm really excited to hear about this and yeah, fascinating study. Uh, yeah, that's what I have to say.
Right. Ali, is there anything you still want to mention, or did we forget something?
No, thank you very much. I think, you know, first of all, I want to thank you for having me. This, as you said, these results are important and also interesting. But when you present it in a scientific way, you know, it may not be very clear for public people. But I think this paper, I tried my best to write it in a very easy to follow. So if people just read the paper, they probably, especially the introduction and also the discussion, is clearly explain what we did and what we found. But what I want to just add that this opens an interesting way to follow up, because in the next step we need to see, okay, what happened to other physiological parameters of human brain, especially for these cognitive performance that are related to non-motor area like prefrontal cortex, and which is more related to cognitive functions. But besides that, I have no more point, and I hope these data and results will be also of use of some policymakers, because honestly, as an evening type, I really don't like to go to school in the morning, but it's too late for me. I mean, I'm already older, you know, for school age. But yeah, when you can learn mathematics better in the evening, why not? Why should go in the morning at 8 a.m.?
Yeah, certainly. So, yeah, that's why I wanted to do this interview, to get this information out there to everybody who's interested. Of course, our channel doesn't have a huge subscriber amount yet, but who knows? Maybe this video will change that. So, yeah, thank you. Michael, do you want to close it off?
Yeah, sure. You know, like, share, subscribe to the channel. And yeah, I guess we'll see you in the next video.
All right. Bye-bye.
Thank you very much. Bye. Have a good evening.